Hello everyone, my name is Morten and I'm a lead scientist at Disease-agnostic, where we develop precision diagnostics for timely and targeted antimicrobial treatment. For infections, time to diagnosis is really critical, and the current gold standard to identify the pathogen in systemic infections, which is blood culture, is insufficient. It has a long turnaround time and a poor sensitivity, leading to an overuse of empirical treatments with broad spectrum antibiotics that are not always effective. That's why we are developing Septrace. Septrace is a metagenomic solution where we sequence pathogen cell-free DNA from the bloodstream and use that to infer the causative pathogen in systemic infections. We've developed a kit that enrich for the microbial DNA. We then subject the DNA to ultra-fast sequencing and then process it with our Scout bioinformatics platform, where we are currently able to provide rapid identification and in time also want to be able to provide genomic antimicrobial susceptibility profile. We believe this can be done in 6 hours from sample in to answer out. The costs after obtaining IVDR we project will be around 800 euros, obviously a lot cheaper for current research use only studies. And then we want to place an automated solution of this on site in the clinical microbiology laboratories to enable as fast turnaround time as possible. First, the method was evaluated in a cohort in the emergency ward at the Aalborg University Hospital, where we had full concordance with positive blood cultures and identified a relevant pathogen in twice the number of patients. Then we have also evaluated it in several new indications, firstly in vascular graft infections at the Aarhus University Hospital, Department of Infection Medicine. And the study was really interesting because it's a very complex patient group. But basically, from 35 patients, 65 samples were collected, and out of these 65 samples, 43 were positive for one or more organisms. And the top pathogens that we identified were classical VGI pathogens such as Staphylococcus aureus, Enterococcus faecium, Candida albicans, and E. coli. As a comparator, concurrent blood cultures from these 65 patient samples were positive in only 3 cases, where 1 was a likely contaminant. There was also an interesting case that if you're interested, I can talk more about that, where we found Epstein-Barr virus in a patient where that was post-mortem diagnosed with large B-cell lymphoma, positive for Epstein-Barr virus. And we found that in 4 consecutive samples, 1.5 years prior to the patient dying. Then we also have proof of concept in pediatric Lyme disease and pediatric bone and joint infections, but maybe there the sensitivity was a bit hampered by it being on biobank samples that were quite old. And then to the team, we are a team of scientists. Some of us are only scientists, and some of us have also background in the corporate world besides that. Then to the ODIN participation, we see ourselves contributing with the Septrace assay, for which we have quite strong clinical data now. It's available as center-way researchers only, now moving towards IVDR, and happy to see that used in new clinical studies and new indications, particularly interested in intracardiovascular infections such as infective endocarditis, and also open to discuss pilot size testing our Septrace assay. So thank you very much.